Thank you so much for the opportunity to speak with all of you virtually. And I'm just going to go ahead and share my screen here. Okay, thank you. Yes, thank you again for the opportunity. Hello from California. And I'm going to be talking about the invasive shot hole borers, mostly what we understand from our perspective of what we've experienced here and what we know from California. And I just want to acknowledge that this research and what we know is part of a multi-campus and multi-agency collaborative effort to develop essential building blocks for integrated pest management across the state. This coordinated network of entities that came together to address the issue represents a variety of interests, including conservation, recreation, city planning, as well as agriculture, and fish and wildlife regulation, as well as academia. And so all of these stakeholders have a vested interest in the problem for different reasons. And a lot of the work we've done over the years, both informally and formally, has been an ongoing collaboration to address this issue in California. So I just wanted to start by giving a background. There we go. Just giving you an overview of the complex here. Many of you already know this, but it's always helpful to just start from the beginning. So the complex Fusarium dieback or shot hole borers is caused by the combined effects of specific Fusarium fungal species that are vectored by Euwalacia ambrosia beetle species, which we commonly call or refer to in the United States as invasive shot hole borers, because they are caused by these we have two beetles, morphologically indistinguishable, but functionally, they functionally behave the same, ambrosia beetles. The Prolifica shot hole borer, and then we have the Kuroshio shot hole borer in California. The beetles pack these fungal spores into the special mouth parts called mycangia, where they carry the spores from tree to tree to grow the fungus for food. And the fungus is also a plant pathogen. And depending on the host and the location, the impacts of these little beetles can be very devastating. So, for example, this is a location in Southern California near San Diego, right on the border near Mexico, where this star is. This is a riparian habitat of willow trees, where within 1 year, over 200,000 willow trees were killed in this riparian forest. This is, of course, the worst case scenario I'm presenting to you, but the impacts can be quite devastating. And we see cascading effects of this mortality as well. So this is looking at that same location on the ground. And what we have found after this mortality, 9 months later, castor bean, which is a highly invasive weedy plant, began to encroach into the area, which prevented establishment and recovery of our native willow trees. And these serve as critical breeding habitat for endangered bird species. And so this is the range of where we have confirmed, just to give you some context and some updates as well. This is the range where we've confirmed Fusarium dieback invasive shot hole borers. The ones in Los Angeles, the Prolifica shot hole borer, are associated with Prolifica shot hole borer are mostly in LA. The ones in San Diego are associated with Kuroshio shot hole borer. At least that's where those introductions were. They are starting to overlap a little bit in distribution now. The beetles were introduced to Southern California on 2 separate occasions from 2 separate locations in Southeast Asia, presumably on packing material. Beetles were later detected in monitoring traps north of the leading edge of the infestation. And just recently, in November of this year, the beetle was confirmed on infested sycamore trees in San Jose, which is the first detection 280 miles north of the leading edge of the infestation. And so we know this was Polypicus shot-hole borer. We know this was a separate introduction, but to find this affecting trees and infesting trees this far north is a really big deal, and it's very concerning. And so there's a big effort right now to respond to this infestation. And just to give you some context, as you may know, California is the heartland of agriculture. We grow everything from nut crops to citrus to avocado. And for context, this is the avocado growing region of the state here in green, which overlaps with the infested range of invasive shot-hole borers. Avocado is a reproductive host, which I will talk about a little bit later and what the implications of that are. In the Central Valley of California is where we grow almond and other deciduous tree crops. This is we have confirmed that the fungus can colonize on this host, but we have not been able to confirm, at least in California, that the beetle can reproduce. We'll talk about that a little bit later. But the distribution of at least within the Central Valley, does not overlap within this growing area. So that's just a little bit of context about like our agriculture system and where the beetle is and is not. So this is a really complex, the beetle is a complex, is complicated. It involves two organisms that are obligately, it's an obligate symbiosis, and it's a complex issue. And because it's a multi-land use and multi-stakeholder issue, we quickly realized that we needed to develop a framework that addresses its complexity so that we could take action using whatever knowledge is available at any given time. And so the essential integrated pest management components that we identified include monitoring and early detection, and developing these tools to quickly identify disease-causing organisms and detect new infestations, researching their basic biology and life history, conducting risk assessments to identify which tree populations and forest communities are most vulnerable to their establishment and most important in their spread, and that includes agricultural crops as well. Developing and evaluating preventative and curative biological, mechanical, and chemical control options tailored to the appropriate needs for particular settings. And then, of course, sharing the menu of options and improving IPM strategies with stakeholders in an iterative process. And of course, this flow is not linear. These building blocks are constantly being updated and refined, often at the same time, through constant discussions and reflection to make sure that we're integrating different perspectives and learn from one another as we go. One thing, I obviously don't have time to hit on all of these. I could give an entire talk on the risk assessment work that I've done, particularly with the modeling that I've done in California, and we have a paper coming out on that. But just a couple of things that I want to hit with monitoring and trapping. That one of the things that we've learned is that there's a variety of different traps that can be used. But the biggest thing that's really important is that we use a lure called cruciferol. This class of ambrosia beetle requires this particular lure. And what's really important is that one of the big lessons that we've learned is that it's really important not to place these traps on the trees themselves, especially if they are a reproductive host. And I'll talk about again what that means. because essentially this is a tool for monitoring. It's a tool for detecting, you know, it's a tool for detection. It's a tool for, detecting and monitoring flight activity or propagular pressure, that sort of a thing. However, if oftentimes what we've seen in California is, when the traps are being hung on trees, you're essentially baiting the beetle. You're baiting the tree itself, and you're creating the tree is essentially the trap, and you're attracting the beetle to the tree and creating an amplifier tree, and creating this kind of source of propagular pressure on the tree. And that tree is essentially a source of spread. So that's just, it's really important to place these traps within 50 meters of the host. So that's just a really big lesson that we've learned. The other thing that I'm just going to really harp on is in this talk is about hosts. Because this has important implications for management. The beetle pathogen complex has a broad host range of 77 tree species that support beetle reproduction, and we refer to those as competent host species. 20 of those competent host species are trees that are native to California and dominate the landscape. Avocado is an important competent host because it produces 90% of the US domestic crop, and anywhere from 25 to 60% of all individuals planted in the urban landscape in Southern California are competent hosts. And so in order to understand this variation, we understand that there is variation. Let me go to this slide. We observed a lot of variation in the severity of the impact, where some hosts just showed some branch dieback and others were killed when they were attacked. And so to understand this variation across the different species, we dug into the biology a little bit more. And we found that when the beetles try to attack a tree, it could not establish a gallery and successfully colonize on some species. While for others, the beetles couldn't establish a gallery, but the pathogen could colonize on the tree itself. So you had the successful colonization of the fungus only. And then on other species, a beetle could bore onto the trunk, produce a gallery, establish a fungal garden, and feed on, make their brood, and then spread onto another host. So both the beetle and the fungus must be able to reproduce for a host to be competent. And each of those steps have the potential to differ among susceptibility in species, and that has really important implications for management. And these tree species are distributed across major kinds of systems, and and so how those are distributed across a landscape matters a lot. So they're distributed across native plant communities, urban forests, and agriculture systems, in our case, in avocado groves. And because they are distributed over this complex landscape, if you know something about the kind of hosts that they are, it might suggest that certain control measures might be better than others, and how you're going to control is going to be important. So, for example, let's just say this is an orchard of a non-competent host, but let's say it is where the fungus can only colonize. So the fungus can colonize, but the beetle cannot reproduce. And let's say immediately in that vicinity, you have surrounding competent hosts. Those competent hosts are going to be a constant source of propagular pressure that can rain beetles into that orchard and cause all sorts of pressure, propagular pressure, and all sorts of other problems that we can talk about. On the other hand, let's say that this orchard is full of competent hosts, but they don't die. that's going to be a different kind of management problem where you might have to be managing lots of branch cankers. And this is the case where we see with avocado. This is a tree species in Disneyland, where this is an example where we found it is a non-host where the fungus could colonize under normal circumstances. But because at this particular time, there was so much beetle pressure in Disneyland Park, it was heavily infested at that time. It became a reproductive host because it was getting so much propagular pressure of beetles constantly attacking the tree. And so this could be a potential outcome. Some of those tree species, some of those tree individuals that are not reproductive hosts could become reproductive eventually if they are getting enough pressure from the outside. And not only can they be getting that pressure, but the beetles themselves can be creating infection cords from and be a source of contamination of other secondary pathogens. So this is looking at the surface of one of those beetles, and the mycelia of a particular canker pathogen that we've seen over and over again colonizing, for example, avocado. And these canker pathogens we've seen in the Botrytis fariaceae actually can get into these entry holes that are carried on the surface of the beetles, and actually can grow more aggressively because the fusarium is a weak pathogen, but these canker pathogens actually grow more aggressively and more quickly through the plant and can actually cause more damage. So this is a case where you may not have a reproductive host in your orchard, but you might be managing for a lot of canker pathogens if you're getting lots of propagular pressure coming in as well. And this is just showing where we've confirmed in a lot of those cases where we've gotten different Botrytis fariaceae pathogens in addition to fusarium that have colonized these tissues. So a lot of the management where this is, I want to just really harp on the fact that managing this beetle, there is not a one magic bullet here. And it really depends on the context. And one of the things that we developed for the urban environment is a management matrix that could be adapted and tailored to agricultural settings. And so some of the factors that we've considered, at least for the urban environment, involve the host species, whether it's reproductive or not, infestation level, how advanced is the infestation, the host value, whether it's a high economic or cultural value host or individual tree, whether it poses a hazard. And some of these factors might differ for an agricultural setting. And this is basically what it looks like when you put it into the matrix and the different action items that one would take. So I'm not expecting you to read the fine print here, but essentially, say if you had an avocado orchard, you're going to want to be managing branches within the grove. If it's a non-reproductive host orchard, you may need to be managing reproductive hosts immediately surrounding the orchard, and monitoring the beetle within that orchard to see if any of your individuals are becoming reproductive or if they're causing any additional cankers, that sort of a thing. But this is something that can be tailored to those settings. And those actions might involve chemical injections, depending on the context. It might involve sanitation, like removing infested branches, chipping, removing entire trees. It really does depend on the context. So there's not a one, you know, there's not one magic bullet. And so just to hit on control options very briefly, I'm not going to go into all of them, because like I said, they all depend on the context, but these are some of the things that we've tried. One of the things that I want to just say about the chemical controls is that we have found that they're not really effective on heavily infested trees. In the context, and again, when we're talking about context, they can't, it depends on where you want to put your resources. If you want to treat high value trees, it can be a preventative measure, but you're going to have to be injecting over and over and over again. So usually the trees where chemical treatments can be somewhat effective is on lightly infested trees, but it's really a combination of these things that need to be employed to really manage the pest. And to know whether your treatments are effective, we have ways of monitoring that by, at least with our like painting over holes to see if these, to see whether these holes are still active or not. So that's just kind of a fun way to test to see if, to see if your treatment is effective or not. And so I'm just going to leave it at that. We have a pest note out. Here's a QR code here. And there's so much more that I could talk about, but I'm happy to take questions. And here's my email address, and happy to have this conversation offline as well. So thank you for your time.